Technology, Chandigarh, and pursued postdoctoral research at the University of Chicago and Yale University, USA. His scientific journey spans across some of the world's leading research institutions, reflecting his commitment to excellence and innovation. Dr. Kumar's research investigates two fundamental cellular processes, protein trafficking and neuronal signaling. His work on post-trans-Golgi network protein trafficking aims to uncover how cells direct specific proteins to lysosome-related organelles, LROs, and dense core granular DCGs, key structures for regulated secretions. Using the unicellular model tetrahymena, his team studies the mechanism of protein targeting and organelle biogenesis, offering insights that may translate to animal systems and improve understanding of diseases. In parallel, in parallel, his research on the major brain G protein, G alpha O, focuses on understanding its role in neuronal development and serotonin mediated neurotransmission. Through transgenic C. elegans models, immunoprecipitation, and mass spectroscopy, his group identifies and characterizes downstream effector interacting with activated G alpha O and examines the conservation of these interactions in mouse brain proteins. These findings aim to elucidate how G protein signaling regulates brain function and contribute to understanding the molecular basis of depression, schizophrenia, and neuropsychiatric disorders. On behalf of the Department of Biotechnology, I request Professor Naveen Kumar Vishwakarma to welcome him with the plants. On behalf of the organizing committee, faculty members, and all participants, I extend a very warm welcome to you, sir. I, we are truly honored to have you with us today, and we are eagerly look forward to your enlightening session.
So first of all, I would like to thank Dr. Santosh for inviting me here and give me platform to discuss my lab research at Bilaspur in this university. So my lab is working on G-alpha O mediated signaling pathway using C-ligand model system. And I would be happy to discuss this signaling pathway with you today. So in our brain, neurons communicate with each other via neurotransmitter, and every neurotransmitter signal through G proteins via G protein coupled receptor. This signaling activate G protein, which further activate downstream effector molecules, which regulate multiple signaling pathway, including neural activity and gene expression. And defect in this type of signaling underlie depression, Parkinson, and many other brain disorder. And we are trying to discover effector protein for major brain G protein, G alpha O. So, can put here this. Okay. But how G protein works? So heterotrimeric G proteins contains 3 subunits that are alpha, beta, and gamma, of which alpha binds with JDP. And this JDP bound G alpha is inactive form. And inactive form binds with receptor, that is GPCR. When ligand such as neurotransmitter or neuropeptides bind with receptor, is allostatic change receptor conformation, which exchange GDP with GTP. So with this exchange, G alpha GTP is dissociated from beta gamma dimer. And this GTP bound G alpha is active form. So activated G alpha and beta gamma dimer then stably bind, form the complex with their affected molecules and regulate multiple signaling pathway. After activation, G alpha protein is inactivated by RGS proteins, which binds with G alpha GTP and hydrolyze G alpha GTP into G alpha GDP. And G alpha GDP again form heterotrimer with beta gamma dimer, binds with receptor and ready for new cycles. So there are four major type of G alpha proteins that are G alpha S, Q, 12 and G alpha O. Downstream affected molecules for G alpha S, Q, 12 are well known. G alpha O is most abundant G alpha protein in our brain. And this G alpha protein receives the signal from all the non-neurotransmitter, but who receives the signal from G alpha O is unknown. So we are trying to discover affected molecule for major brain G protein G alpha O. So since 35 years, many labs are trying to discover affected molecules for G alpha O by using different methods. And some have found some binding proteins, but no convincing effector. So question is rise, does G alpha O have any effectors? So one possibility is that G alpha O could serve to release G beta gamma. And G beta gamma is responsible for downstream signaling. But this idea is refuted by C ligand genetics because in C ligands, knockout of G alpha O cause loss of signaling and or expression of G alpha O leads constitutive signaling. So these genetic evidence in C ligands show that G alpha O must have effector. So we are trying another approach to discover the affected protein of G alpha O. For this purpose, we immunopurify G alpha O affected protein complexes from the brain, and then we identify G alpha binding protein by mass spectrometry. So G alpha GTP is active form. Active form stably bind from the complex with affected molecules. G alpha GDP is inactive form, and there is no interaction between inactive G alpha with affected molecules. So G alpha proteins binds with their affected molecules only in the active state, but not in the inactive state. So generally 1% G alpha O present in the active state in our brain, and 99% G alpha O remain in the inactive state, and only activated G alpha O from the complex with affected molecules. So if we pull down G alpha O, there is only 1% chance to pull down G alpha O affected complexes. So we wanted to efficiently pull down G alpha O affected complexes. For this purpose, we generated constitutive active mutation in G alpha O. We substitute 205 glutamine amino acid with leucine. With this mutation, G alpha O GTP is not hydrolyzed into the GDP. So G alpha O GTP remain in the active state, stably bind from the complex with affected molecules and continue to amplify signal. So we used this gain of function mutant in our study to efficiently pull down G alpha O affected complexes. So we injected this mutant G alpha O GFP plasmid with most SCI transposes into the C ligands animal, which carry only one most SCI recombinase site in their genome. So only single copy integrate into the genome. Then we screen the animal for most SCI insertion, and then we confirm integration of single copy of mutant G alpha O GFP into the genome by genotype PCR, and then we selected single homozygous animal which express mutant G alpha O GFP. So next we check the expression of mutant G alpha GFP by Western blotting using anti-G alpha antibody. For the control, we use the animal which express single copy of wild type G alpha GFP. So here we can see expression of mutant and wild-type GLPA-GFP is very similar, and level of expression is comparable to endogenous GLPA-O. So there is no any artifact or expression of GLPA-O, so we can use this mutant GLPA-GFP for pull-down assay to identify binding protein and affected molecule for GLPA-O. Next, we check whether this mutant GLPA-GFP is in the active state or not, because only activated GLPA-O from the complex with affected molecules. So GLPA signaling regulate locomotion and egg-line activity in C. ligands. So we check locomotion and egg-line phenotype in mutant animals. So first we check locomotion phenotype. So here we can see this is wild-type C. ligands, which is moving with deep body band, and this is mutant animals. So mutants are slow moving with shallow body bands compared to wild type one. And this is expected phenotype of gain of function mutant of GLPA-O. Next, we look for egg line phenotype. So GLPA-O signaling inhibit egg line in C. ligands. So wild type animal generally keep only 10 to 12 egg inside the uterus. And here we can see in mutant animal, GLPA activity is increased. So we are seeing many egg inside the uterus, and this is also expected phenotype of gain of function mutant of GLPA-O. So both locomotion and egg line phenotype suggesting that GLPA-GFP, mutant GLPA-GFP is in the active state. We further confirm it by biochemical method. So in biochemical method, we pull down GLPA-O from wild type and mutant animal, and after pull down, we look for beta subunit to get the idea about the activation of GLPA-O. So beta subunit binds with alpha only in the inactive state, that means heterotrimeric form, but not in the active state. So if we see beta subunit, that means GLPA-O contain inactive fraction of GLPA-O present in the animal. But if we not see beta subunit, that means GLPA-O is in the active state. So here we pull down wild type and mutant GLPA-GFP by anti-GFP antibody, and we detected both wild type and mutant GLPA-GFP by Western blotting using anti-GLPA-O antibody, and then we look for beta subunit to get the idea about the activation of GLPA-O. We can see we detected beta subunit in wild type GLPA-GFP pull down. That means wild type animal contain inactive fraction of GLPA-O, but in mutant GLPA-GFP pull down, beta subunit is completely absent, which further confirm that this mutant GLPA-GFP is in the active state. After this confirmation, we pull down this GLPA-O from C. ligands lysate and check the sample on the gel system. We can see this is no transgenic pull down sample, and this is wild type GLPA-GFP pull down sample. So in wild type GLPA-GFP pull down sample, GLPA-O remain in the inactive state. So we detected all three subunit, alpha, beta, and gamma. But in mutant GLPA-GFP pull down sample, GLPA-O remain in the active state. So we only detected beta subunit, but we only detected alpha subunit, beta and gamma both are absent. But unfortunately, we did not see any unique protein band in the activated GLPA pull down sample. It may be due to high background signal. So we also perform alternative approach to pull down GLPA-O and affected complexes. For this purpose, we use wild type C. ligands lysate, which mostly contain inactive fraction of GLPA. We incubated wild type C. ligands lysate with GTP gamma S. GTP gamma S is non-hydrolyzing analog of GTP, which binds with alpha subunit, but it is not hydrolyzed into the GDP. So G-alpha-O GTP gamma S remain in the active state, binds with receptor, and form a stable complex with affected molecules. We pull down these complexes, and then we look for beta subunit to get the idea about the activation of G alpha O subunit. So here we can see in presence of GDP, G alpha, we detected beta subunit, that means G alpha O remain in the inactive state. But in presence of GTP gamma S, inactive fraction change into the active fraction. So we are not seeing beta subunit here. We perform the same assay with mouse brain lysate because there is 80% sequence identity between C ligands and mouse G alpha. So we expect that affected will be also conserved between these two model systems. So we in vitro activate G alpha O in mouse brain lysate, and then we fractionated mouse brain lysate proteins by gel filtration column, and then we collected different fraction and perform Western blotting using anti-G alpha antibody. So expected size of inactivated G alpha O is 82 kilodalton and activated G alpha O is 40 kilodalton. So we can see both inactivated and activated G alpha O from the big complexes in the brain. So here we wanted to remove non-specific protein in pull down sample. So here we only pick G alpha O positive fraction, and then we perform pull down using anti-G alpha antibody, and then we check the sample on the gel system. Here, this is control antibody pull down sample. This is G alpha pull down sample in presence of GDP. So in presence of GDP, G alpha O remain in the inactive state. So detected alpha, beta, and gamma, all three subunits. But in presence of GTP gamma S, inactive fraction change into the active fraction. So we only detected alpha subunit, beta, and gamma, both are absent. But still we do not see any unique protein band in the activated G alpha pull down sample. So initially we thought we will get some unique protein band, we will cut that band and then process those bands for mass spectrometry analysis to identify binding protein and affected molecule for G alpha. But in this situation, we perform mass spectrometry analysis with whole length. So by using mass spectrometry analysis with whole length, we found 646 protein in control antibody pull down sample, 1,461 proteins in G alpha pull down sample in presence of GDP, 1,258 protein in G alpha pull down sample in presence of GTP gamma S. We repeated the immunopurification and mass spectrometry using worm lysate activating G alpha O with mutant Q205L or with GTP gamma S. So in our mass spectrometry data, first we check whether previously known G alpha binding protein are present in our data or not. So here's all known G alpha binding protein were detected in our mass spectrometry data. And then we analyze for similar mass spectrometry signals that reproduce in replicate and different version of the experiment. On the basis of it, we found 129 new G alpha binding proteins. And out of 129 candidates, one half were conserved in both mouse and worm. And 1/3 are proteins involved in neuronal signaling. But still there are so many candidates. So question is how to sort out these candidates. To answer it, we applied second filter. We selected this candidate and we knocked down them in C ligands, and then we look for knockout G alpha O phenotype. Then we combined both filters together and then we found at least 30 candidates. But still there are 30 candidates. So question is how to validate these candidates. To answer it, we pull down G alpha O from mouse brain lysate, and then we perform Western blot with antibody to candidate binding protein and reprobe with antibody to G alpha. So by using this method, I'm going to show you and successfully validate G alpha binding proteins, that is chem kinase 2. So here we can say we pull down G alpha O and detected chem kinase 2 using specific antibody against chem kinase 2. And we can see here chem kinase 2 is present in input sample. Chem kinase 2 is completely absent in control antibody pull down sample. but we detected chem kinase 2 when we pulled down G-alpha O using anti-G-alpha O antibody. And now I'm going to show you one failed to validate G-alpha binding protein, that is endophyllin 1. We can say we detected endophyllin 1 in input sample, but it is completely absent in pull down sample. And with this, we did this for 30 candidate G-alpha binding proteins. And we found 9 novel G-alpha binding protein. Now we are checking potential effector for G-alpha O. So G-alpha O signaling inhibit neurotransmitter release, and another G-alpha proteins, that is G-alpha Q, which activate neurotransmitter release. So G-alpha O and G-alpha Q oppose to each other. And this model diagram is showing how G-alpha Q signaling activate neurotransmitter release. And recently we discovered some new G-alpha O binding proteins that are sphingosine kinase, diacylglycerol kinase gamma, synaptogenin 1, and chem kinase 2. So these candidates make sense as a possible effector via which G-alpha O could oppose G-alpha Q signaling. And one candidate is highly evident in the brain, like a G-alpha O, that is chem kinase 2. So chem kinase 2 is calcium and calmodulin dependent protein kinase, which is autophosphoride and then phosphoride, other substrate. It is also reported that chem kinase 2 and G-alpha O knockout mutant in C. elegans show many shared phenotypic defects such as locomotion and egg-line defect. And double mutant study suggests chem kinase 2 acts upstream of G-alpha O to activate it, but there is no any biochemical evidence about it. So we wanted to check whether G-alpha O is activated by chem kinase 2. For this purpose, we prepare the lysate from wild type and chem kinase 2 gain of function worms, and then we pull down G-alpha O, and then we look for beta subunit to get the idea about the activation of G-alpha O. So first we check the expression of beta subunit in wild type and chem kinase 2 gain of function animal. We can see expression of beta subunit is very similar in both animals. And then we look for beta subunit level in the pull down sample. We can see very less amount of beta subunit is present in chem kinase 2 gain of function animal. That means more G-alpha O is present in the active state, and this activation is due to chem kinase 2. So chem kinase 2 is calcium dependent protein kinase, which autophosphoride and then phosphoride other substrate. So we ask the question whether G-alpha O is phosphorylated by chem kinase 2. To answer it, we purify G-alpha O, chem kinase 2 alpha, and calmodulin protein in bacterial system. And then after purification, we perform in vitro phosphorylation assay using gamma P32 ATP. We can see purify chem kinase 2 phosphoride G-alpha O in vitro condition in presence of calcium and calmodulin. So here's G-alpha O is phosphoride by chem kinase 2. Then we look for potential phosphosite in G-alpha O protein. For this purpose, we perform the same assay with non-radioactive ATP, and separately we pull down G-alpha O from mouse brain lysate using anti-G-alpha antibody, and then we perform mass spectrometry analysis to identify potential phosphosite in G-alpha O, which is phosphorylated by chem kinase 2. In both cases, we found threonine 48 was phosphorylated in G-alpha O by chem kinase 2. And now we are checking how this phosphosite in G-alpha O affect G-alpha O function. So this is a story about G-alpha signaling pathway. My lab is also working on another pathway, that is lysosome-related organelle biogenesis. We are using protozoan tetrahymena thermophila as a model system to study this pathway at molecular level. So I will give very brief introduction about this project, and then I will show you some new result about this pathway. So release of signaling molecules from cells such as neurotransmitter from neurons, hormone from endocrine cells, and immune peptides from immune cells is a key mechanism for intercellular communication and tissue coordination. These signaling molecules are released from the cells by fusion of signaling molecule containing vesicles with plasma membrane. And this vesicle is called secretory granule. After fusion, secretory granule release is contained outside the cells upon stimulation. And secretory granules are the products of the secretory pathway. In this pathway, newly synthesized proteins first transport to ER, then pass through Golgi compartment and a transphase called trans-Golgi network, and then sorted into variety of different compartments such as secretory granule, endosome, lysosome, and lysosome-related organelles. And we are working on lysosome-related organelle, and I will only discuss here about LRO-biogenesis pathway. So lysosome-related organelles are derived from an interconnected network of endocytic and secretory pathway. And these are the unique component of LRO. So LRO are synthesized by some specific cell type, and they perform some specific function. For example, lytic granule, which stores hydrolytic enzyme. And lytic granules are synthesized by T lymphocyte cells, which kills virus-infected and cancerous cells. So that means lytic granule only synthesized by T lymphocyte cells, and they perform some specific function. Okay? So these are the example of LRO in different model system. And recently, many labs added so much new information about this biogenesis pathway, but still this biogenesis pathway is not well understood in animal cells. So we look for alternative simple model system to study this pathway at molecular level. So recently my group has established that prominent secretory compartment, that is mucocyst in Tetrahymena thermophila, which is also LRO because it is derived from interconnected network of endocytic and secretory pathways. So we selected Tetrahymena thermophila as a model system to study this pathway at molecular level. So so this is Tetrahymena cells. It is single cell model system. So Tetrahymena is a freshwater protozoans. It has 25,000 coding genes, and it has two nucleus that are macronucleus and micronucleus, and one Tetrahymena strain can mate with 7 different Tetrahymena strains. So Tetrahymena is an advanced model system because it shares many essential biological pathways with higher eukaryote, including endolysosomal and secretory pathway. In addition, Tetrahymena have many practical advantages such as short doubling time, that is 2.5 hours in inexpensive media. It is non-pathogenic and large size, which is suitable for microscopy. Tetrahymena can be stored long-term in the liquid nitrogen. It is easy to knock out gene in this model system, and gene can express under constitutive and individual promoter. So these tools make powerful genetic, molecular, and cellular analysis possible in this simple ciliate. And many notable discoveries have made using this model system, including structure of telomerase enzyme, early RNA splicing, histone acetylation, and discovery of catalytic RNA ribosome. So all these discovery made using Tetrahymena thermophila. So now I'm coming back again on LRO biogenesis pathway in Tetrahymena thermophila. So Tetrahymena mucocysts are formed by two classes of proteins. One is granule latex protein, second is granule tip proteins. And only granule latex protein undergo proteolytic processing, but not granule tip proteins. Tetramycosis can be visualized by indirect immunofluorescence assay using anti-GIAN antibody. Here we can see we stain wild type cells with anti-GIAN3 antibody to visualize mucocyst. In cross section, we can see elongated dark mucocyst at plasma membrane, and in surface section, we can see linear array of dark mucocyst at cell surface, and all dark mucocyst exocytosis within the second upon stimulation. So GIAN proteins are synthesized as a proteins and then proteolytically processed in post-Golgi compartment, that is immature mucocyst. So inside the immature mucocyst, pro-GIAN proteins which are changed into the mature GIAN protein by key processing enzyme cathepsin 3, and mature GIAN protein can be detected within the crystallizing core. Are you seeing here? This is something different. Something different pattern. Are you seeing here? So mature GIAN protein can be detected within the crystallizing core as a crystal at level of electron microscopy. So after processing of pro-GIAN proteins, immature mucocyst change into the mature mucocyst, and during this maturation, shape of mucocyst is changed from round to elongated, and then mature mucocyst dock to plasma membrane, fuse with plasma membrane, and release its content outside the cells upon stimulation. So proteolytic processing of pro-GIAN protein is closely linked to core formation of mucocyst. If there is defect in processing of pro-GIAN protein, then we will see defect in mucocyst formation. So now key questions are, how do cells organize and regulate this complex pathway? And what are the factors involved in regulating compartment-specific assembly of LRO? To answer it, I took the advantage of an online available database, that is TetraFGD, in which expression profile of all tetramina gene can be viewed. By using this database, I learned that mucocyst genes are highly co-regulated under variety of conditions that are growing, starvation, and conjugation condition. So if we know the one gene of this pathway, on the basis of expression profile, we can get the information about all the interconnected gene of this pathway. So by using expression data, I found around 1,000 genes which are highly co-regulated with non-mucocyst associated gene. But there are 1,000 genes, so question is how to sort out these genes. To answer it, we applied second filter. For this purpose, we treated wild-type cells with dibucan. So dibucan is secretagogue, which remove all mucocysts from cells. Okay, and then we again grow the cells in normal media, and then we look for the genes which are highly upregulated during new mucocyst formation. That means those genes may be involved in mucocyst biogenesis pathway. Then we applied both filters together, and by screening integrate data set, we identify new candidate of mucocyst, and these candidates are also show high homology with higher eukaryotes. And some candidates we already published in Journal of Cell Biology, Current Biology, Molecular Biology of Cells, and Eukaryotic Cell journals. And this is current model for mucocyst biogenesis in Tetramina thermophila. We can see two vesicles bud from endosome and TGN. One vesicles contain pro-GIAN protein, and another vesicles contain syntaxin. Sorry, the syntaxin is another protein present in the same vesicles. So another vesicles which contains sortilin receptor and its ligand cathepsin 3 GRT1 and IGRT1, and cathepsin 3 is key processing enzyme which process progeral protein. So you can say enzyme and substrate both are remain in separate vesicles. Okay. These vesicles also contain RAB7, which is marker for late endosome, and VPS88. Both vesicles fuse to generate immature mucosis. You can see chiral proteins and cathepsin both are present in these vesicles. So immature mucocyst then change into the mature mucocyst, and this maturation is dependent on adaptive protein 3. Mature mucocyst fuse with plasma membrane and releases content outside the cells upon stimulation. So inside the immature mucocyst, prochiral proteins change into the mature chiral protein, and this processing is dependent on cathepsin 3. Cathepsin 3 is proteolytic enzyme, which is enzymatically active in acidic conditions, and that acidic conditions are generated V-ATPase complexes. So basically we were looking for key factor for processing of prochiral proteins. Okay, so one key factor is acidic pH, and this acidic pH is generated by V-ATPase complexes. So we will look for V-ATPase complex which is associated with mucocyst in tetrahymena thermophila, and we found 18 V-ATPase gene in tetrahymena thermophila, and out of 18 V-ATPase gene, only single A1 subunit which is co-regulated with non-mucocyst associated genes, that are GRL1 and GR3, and that subunit is also upregulated during new mucocyst formation. So we selected this V-ATPase subunit for further study. So first question we ask whether this subunit is localized to mucosis. To answer it, we perform live cell microscopy with cells co-expressing V-ATPase 2X amnion and mucocyst marker GRL3 3X mcherry. We can see this V-ATPase 2X amnion completely co-localized with mucocyst marker, which confirm that this A1 subunit is localized to mucocyst. And we also confirm expression of full-length V-ATPase 2X amnion MYC protein by Western blotting using anti-MYC antibody. So this A1 subunit is localized to mucocyst. So next question we ask whether this subunit is involved in mucocyst biogenesis pathway or not. For this purpose, we knock out this gene by homologous recombination method in tetrahymena thermophila, and then we confirm knockout of gene by RT-PCR, and then we perform exocytosis assay. So in this assay, when we treat wild type cell with secretagogue, diobecan, mucocyst container release is a macroscopic protein aggregate which form thick flocculent layer. So you can see after diobecan treatment, wild type cells which form thick flocculent layer, that flocculent layer was completely absent in knockout V-ATPase cells. That means this subunit is involved in mucocyst biogenesis pathway. But there are two possibilities. Mucocysts are formed, but mucocysts are not exocytosis. Second possibility is that mucocysts are not formed. So we look for this possibility. So we visualize mucocyst in wild type CU428 cells and knockout V-ATPase cells. So we visualize mucocyst by anti-GRL3 and anti-GRT1 antibody. We can see mature mucocyst which dock to plasma membrane in wild type cells, but we do not see mature and dock mucocyst in knockout V-ATPase cells. And most of GRL3 and GIT signal trapped inside the cell, and this aberrant structure may be represent intermediate of mucosis biosynthesis pathway. And this result is consistent with electron microscopy result in which we did not see mature and dark mucosis in knockout VATP cells. So this result suggests that VATP's A1 subunit is required for mucosis formation. But now question is, why A1 subunit is required for mucosis formation? So we investigate why actually A1 subunit is required for mucosis formation. For this purpose, we analyze cell lysate from wild type and knockout VATP cells, and then perform Western blotting using anti-GRL1P and anti-GRL3P antibody. We can see pro-GRL proteins which are completely changed into the product in the wild type cells, but in knockout VATP cells, overaccumulation of pro-GRL proteins which are not changed into the product. And processing of pro-GRL proteins are closely linked to core formation of mucosis. So here in knockout VATP cells, processing of pro-GRL protein is so pro-GRL proteins are not properly processed in knockout VATP cells. So we are seeing defect in mucosis formation in knockout VATP cells. Is it clear? So now again question is, why A1 subunit is required for processing of pro-GRL proteins? So VATP is H plus pump, which pump H plus ion into the specific compartment and generate acidic environment. That acidic environment may be activate key processing enzyme, and key processing enzyme then process pro-protein into the mature proteins. So that means that compartment should be acidic. So we check whether mucosis is acidic or not in Tetrahymena thermophila. So we tag mucosis protein GRL3P with pH lurine and M-cherry. pH lurine is sensitive to pH and so only fluorescence when pH is higher than 6. Another tag, that is M-cherry. M-cherry is not sensitive to pH. So one tag is sensitive to pH, but another tag is not sensitive to pH. So we express this, endogenously express GRL3P, pH lurine, M-cherry in wild type cells, and then we look for pH lurine and M-cherry signal. If we see pH lurine and M-cherry signal together, okay, that means that compartment is less acidic. Less acidic, that means pH should be higher than 6 or non-acidic. But if we say M-cherry signal and pH lurine signal is absent, that means that compartment is acidic. Okay, so first we check pH lurine and M-cherry signal in mature and dark mucosis. We detected both pH lurine and M-cherry signal in mature and dark mucosis, which is completely overlay with each other. That means mature mucosis is less acidic or non-acidic. Then we look for same signal in non-cortical compartment of mucosis, that means in cytoplasmic pool of mucosis, which represent intermediate of mucosis. Okay, so most of M-cherry fluorescence puncta do not contain pH lurine signal. That means those compartments are acidic. We further confirm this result by treating GRL3 pH lurine M-cherry cells with buffalomycin A. Buffalomycin A disrupts VATPase activity. So if VATPase activity is disrupted in intermediate of mucosis, that means we should see pH lowering signal, and that pH lowering signal should be co-localized with M cherry. So here we can say after treatment, BFA treatment, we can say we detected pH lowering signal which is completely co-localized with M cherry. That means intermediate of mucocyst are acidic compartment. There is no quenching effect. Okay. So mucocyst is not stably acidic in Tetrahymena thermophila. Intermediate of mucocyst are acidic, and mature mucocyst is less acidic or non-acidic. So then we directly measure pH inside the mature mucocyst and intermediate of mucocyst. For this purpose, we treated cells with inophore, valinomycin, and nigricin, okay, in different pH buffer. So these inophore form equilibrium in intracellular and extracellular pH. If you keep cells in pH 5 buffer with inophore, then pH inside the compartment and outside the cells is same. Okay, if you use 6, then inside the pH in lysosome should be 6 also. That means you can manipulate pH inside the compartment by using these inophore. They basically exchange K plus ion with H plus ion. They input K plus ion and replace K plus ion with H plus ion. So we treated cells with inophores in different pH buffer, and then we measure M cherry to pH lowering ratio at different pH buffer in presence of inophore and generated standard curve. For the test condition, we keep the cells in neutral pH buffer, pH 7, without inophore, and measure the same M cherry to pH lowering ratio in mature and dark mucocyst and intermediate of mucocyst. So we found pH 5.6 in intermediate of mucocyst, which is acidic, and we found pH 7.2 in mature mucocyst. That means it is non-acidic. Now question is, why intermediate of mucocyst are acidic and mature mucocyst is non-acidic? So V-ATPase complex are formed by two domains. One is V1 domain and second is V0 domains. That both domains form hollow complex. Okay? And hollow complex is enzymatically active. Enzymatically active, that means it can pump H plus ion into the compartment and generate acidic environment. But in many cases, this complex is disassembled. So incomplete complex cannot maintain acidic environment. So it is possible that this hollow complex is present on intermediate of mucocyst, and this hollow complex generate acidic environment in intermediate of mucocyst. So intermediate of mucocyst are acidic, but during the maturation process, it is possible that this complex is break down. So mature mucocyst is non-acidic. So we look for this possibility. So we check co-localization of V1 and V0 subunit with mucocyst marker in intermediate of mucocyst and also in mature mucocyst. So this is VMA1. VMA1 is subunit of V1 domain. We can say this VMA1 is co-localized with mucocyst marker GRL3. Okay, so that subunit is present in intermediate of mucocyst. We also check two other subunit of V1 domain that are VMA8 and VMA10, and these two subunits also present in intermediate of mucocyst. Then we look for V0 domain subunit, that is A1P. So A1P is also co-localized with mucocyst marker GR3P in intermediate of mucocyst. So V0 and V1 subunit all are present in intermediate of mucocyst. Then we check whether those subunits are transported to mature mucocyst or not. So here we can see A1P, which is part of V0 domain, transport to mature and dock mucocyst, but VMA1, VMA8, and VMA10 all are absent in mature and dock mucocyst. That means this hollow complex is present on intermediate of mucocyst. So intermediate of mucocyst is acidic, but during maturation process, this complex is disassembled. So incomplete complex cannot maintain acidic pH in mature and dock mucocyst. Now, the question is, who regulate this disassembly? So by using mutagenesis analysis, we found adapted protein 3, which require for disassembly of VATPs complex. We can see expression of GRL3P pH lowering M-cherry signal in knockout AP3 cells. Most of M-cherry fluorescence puncta do not contain pH lowering signal. That means those compartments are acidic. If those compartments are acidic, that means hollow VATPs complex is present on those compartments. So in the absence of AP3, VATPs complex is not disassembled. Now we are checking how this AP3 regulate disassembly of this complex. And on the basis of this study, we updated model for mucosis biogenesis pathway in Tetrahymena thermophila. We can see two vesicles bud up from TGN and endosomes. Both vesicles fuse to generate immature mucocyst, and this fusion is dependent on VATP's A1P subunit. And immature mucocyst is acidic because hollow complex of VATP is present on immature mucocyst. But during maturation process, this complex is disassembled, so mature mucocyst is non-acidic. And this disassembly is dependent on adapted protein 3. So my lab is collaborated with various labs inside the India and outside the India, also well funded by different funding agency, UGC, DBT, DST, India Alliance, Wellcome Trust, DBT Intermediate Grant, and intramural funding from BRIC and CCS. Thank you very much. So I don't know you understand all this data or not, but I try to tell you how to do science, how to, if you ask, when you discover something, then analyze your data and ask the question, why you are seeing this defect. अब सबको समझ में आए कि ये So this work was done by my student. It took around 3.5 years. तो 3.5 years का सारा काम जो है एक मॉडल के अंदर आ गया उसमें, एक स्लाइड के, बट उसके लिए बहुत ज़्यादा मेहनत लगती है. तो मेन चीज़ है आप लोगों के कोर्स के अंदर कि किस तरह से आप नेक्स्ट लेवल पे जाके टेक्निकल पर्सन बनने की जगह साइंटिस्ट बनने का ट्राई कि व्हाई यू आर सीइंग एक्चुअली those phenotype and how do you design experiment to address those questions. तो questions
जो जी अल्फा ओ इफेक्टर कॉम्प्लेक्स हम उसको एज अ थेरेप्यूटिक टारगेट कैसे यूज कर सकते हैं न्यूरो डिजेनरेटिव डिजीजेस
कर रहे हैं लोग ऑलरेडी. एक्चुअली इसके कारण जो है मूवमेंट डिसऑर्डर होता है. तो मूवमेंट डिसऑर्डर के लिए लोग देख रहे हैं कि जो सिग्नलिंग पाथवे है अफेक्टेड मॉलिक्यूल्स नहीं है, तो लोग ट्राई कर रहे हैं कि इसी पाथवे को बाईपास करें। इसकी जगह यदि इसका अफेक्टर मिले तो आपका अपस्ट्रीम जो सिग्नलिंग पाथवे है उसको एक्चुअली कोई उसका फिर जो है आपको कुछ भी जी अल्फा एब्सेंट भी है, नॉकआउट है, म्यूटेंट है, उसके बाद भी सिग्नलिंग पाथवे होगा इफ यू प्रोवाइड अफेक्टर मॉलिक्यूल्स। मैं अभी तक 4 बजे तक ही, इफ यू वांट टू आस्क एनी क्वेश्चंस देन इसकी intermediate of mucosis. What is criteria for? What is criteria for intermediate of mucosis? So actually, when we do not have good marker for immature mucosis, so someone asked the question, how do we say it is immature mucosis? So mucocysts are continuously actually synthesized. So there means actually there are so many intermediate, and we don't have marker. So collectively we call them intermediate of mucosis. So intermediate mucosis, they are, we can distinguish between intermediate of mucosis and mature mucosis on the basis of shape. Intermediate of mucosis are round shape, but during the maturation, this round mucosis change into the elongated shape. But we do not have the good marker for all intermediates, so we collected for intermediates. तो इसमें ये फाइल डिलीट करना है,
डिलीट करना है।
ये वाला था।
ओके
कैसे डिलीट कैसे करना है?
डिलीट करना है।
हाँ, यहाँ से।
और क्वेश्चन पूछे क्या?
Okay. Okay, working now. I'm sorry. Okay, I apologize. Just this morning, I have unexpected engagement invite. So I went there to meet another group of students, and some sort of delay. I apologize for all the issues. So now we are going to go to the last section for this crosstalk. I'm going to use one example, the P66 protein in the crosstalk, crosstalk regulation and carcinogenesis. Okay, this protein is very unusual. Are we talking about the free radical? Are we talking about, say, oxidase? Are we talking about the reductase? But interestingly, is unexpected. This protein per se is not an enzyme. It actually interacts with the cytochrome C in the mitochondria, and then produce ROS, then involved in growth regulation, involved in carcinogenesis. So again, I try to remind you, biology, as my postdoc, my former postdoc remind me all the time, biology is a lot of crosstalk, a lot of complicated system. Okay, so the topic of, for this 1 week is a multi-omic, right? Multi-omic. It's essentially, it's a link, the gene, the protein, and the metabolite, and also the regulation. So by this, we can understand deeper in the molecular level how the interaction. This will relate to the health and the disease. So that's what I give it subject, a title say multi-omic, because indeed we are not only going to study only one as a genome or the epigenomic regulation. So then for the cancer research, we need to understand the health condition, the normal condition. Why? If we don't know the normal situation, normal physiology regulation, how we know the cell, how they change in the pathology situation, right? So only we understand the physiology, then we know the pathology. So the pathological research, say cancer, is one step ahead further of a normal physiology research, okay? So this is what a lot of challenge for cancer research scientists. You need to know a lot, not everything. You need to learn the basic fundamental regulation. Then you can advance for the cancer research. So for the omic, the gene study we call genomic. For the gene regulation by epigenetic regulation, they call the epigenomic. Or the protein, they call the proteomics. Then it regulates through the phosphorylation regulation. They call the phosphoproteomic, glycosylation, glycomic, redox, they call redoxomic, metabolite, metabolome, so on and so on. So all these are actually a systematic study. And as I mentioned in the beginning, the microbiome. Initially, we saw the microbiome in our intestine, in our GI system, is some sort of independent. We didn't maybe produce some vitamin for our body usage, right? But turn out now it's more than what we know. It's a microbiome in the GI system. Actually regulate our immunity. Okay, regulate our immunity, and not only regulate our immunity, but also regulate our drug resistance. Yeah, so we can transfer the microbiome in the feces from a patient who is drug sensitive to a patient who is drug resistant. Then the later patient, the new patient, become drug sensitive. So you can see how the microbiome is doing, not only the immunity, but also even the drug sensitivity, the chemo resistance. So all this together, they can call the spatial biology. So it's another new term. They say, yeah, the spatial biology. We can include everything. But how can we know so much by one person? It's impossible. So that's why we need a lot of collaborators. Okay, so even now we are talking about the AI, artificial intelligence. So the AI could help us organize, sorting out the data, but the ultimate decision, the judgment call, is in yourself. You are the person make the judgment call. Okay, so the artificial, the AI system, sometimes they give us the false positive result. But we are the person make the ultimate decision, we make the judgment call. So for example, this way back over 20 years ago, we started what we call the functional genomics, and now they call the transcriptome. We actually analyze the mRNA expression. So this functional genomic is, because it's mRNA expression, we call the function. So we have two cells. We have a control, we have experiment one. So this control cell in here is we call the C33 LNCAP cell. Then this is C81 LNCAP cell. Now we can compare after the mRNA hybridization, we can compare the spot. For example, this is much stronger, darker, right? So this one going up. This one seems going down. This one compared seems not much. So this so and so, you can see the change in here. So going down, going up. Yeah, so that's what you got to have a control. Then you know what's going on. So similarly, you have a normal condition. Then you have pathological condition. Then you know what's going on, what's changed. Okay, so for pathological study, you need to learn the physiological phenomena first. So that's one step further. Okay. So I'm going to tell you, use our P66 chick as an example, how we integration of the signal pathway, the free radical, protein phosphorylation, and the steroids involved in the carcinogenesis. So this essentially one protein will do the job. Yeah. So what is a P66 chick protein? What is a gene? They actually has three. The protein has three isoforms called P66 chick, P52, and the P46. All the three proteins encoded from one gene. Okay, so depending on the initiation codon, if they're from here, will be 52 and 46. Then if the second initiation codon here, then will be P66 chick. So why here? Because this one actually truncated, it's non-coding region. So by this way, then we have the protein. This is a P66 protein, 52, 46. And the P66 protein has specific N-terminal domain. So this is a part we can use to produce antibody. We can generate antibody. But unfortunately, for other two study, very difficult to study the P46 because we have no tool, no antibody. Very difficult. So the study of P46 very much behind. So then we spend a lot of time on P66 sugar protein. Then they do have a phosphorylation site. So the phosphorylation at the serine 36 actually go to apoptosis. Okay, the phosphorylation on here is apoptosis. So Dr. Horwitz, who actually discovered taxol in the cancer therapy, he actually used the P66 sugar protein release as a phosphorylation on here, the phosphorylation here, as a biomarker to show whether the taxol can cure this cancer patient, this cancer cell or not. Because if there's a phosphorylation on this site, it go to apoptosis. So this is a biomarker used by Dr. Horwitz, and who actually is a scientist discovered taxol. It's widely used now in the cancer therapy. Yeah. And actually, interesting is we find the P52 chick at the tyrosine 317. It is a site regular androgen action or estrogen action. If we mutated this site 317, a prostate cancer cell will resist to androgen stimulation. then breast cancer cell will not send it to estrogen stimulation. So this site is very interesting, regulate steroid hormone action. So that I mentioned to you earlier is a P66 protein could be considered as a longevity protein. So it's actually in the 1999, when they knock out the gene, one copy of the gene, the lifespan increased. Both copy going down, the lifespan increased 30%. So this is a very substantial 30%. That means if you are expecting life is 70, then that means you can grow, you can survive to 90. Okay, so this is followed very nicely. One copy increase 15%, two copy both knocked down, increase 30%. So people were so surprised and so interesting to see this is a longevity gene. Yeah, they call it a longevity gene. But I don't know what's happening. Dr. Perusi just have a interesting to continue the research. He put those mice in the natural condition. Turn out those mice had shorter life. So the previous one was the mice in the cage, in a well-maintained condition. They have prolonged life. But when they're in the natural environment as other mice, they actually shorter life. Because turn out they regulate in the lipid metabolism, okay? In the food consumption, they actually regulate, involved in the fat accumulation. So they cannot accumulate fat, so they lost their resistance in the cold weather. So in your country, it's so warm, you don't know how cold it will be in the cold weather. For example, in Omaha, in the winter, we can reach to minus 20 Celsius, minus 20 Celsius. So like the freezer in your refrigerator, in your home, okay? So we live in the cold ice chamber, that's minus 20 in the winter. We can reach it there. Yeah. So if we don't have the fat to protect us, the thermal protection, we cannot survive. So that would be the question. For example, you might think, how can we survive? But luckily in US, we drive. We all drive. So we can, actually we have a heavy jacket. Whenever we walk out, we put the heavy jacket on and a short distance walking. We don't walk long distance in the cold weather. No, they actually don't like it to walk in the cold weather. And we have very heavy duty boots and a very heavy duty jacket. You even cannot think of that, okay? Yeah, so when the mouse is similar to a human being, we don't have the fat, we don't have the lipid for protection. We cannot survive in cold weather. How about in human? So in human, actually they got the fibroblasts from the skin and from different age. Turn out is the skin, the P66 sheet protein at the centenarian actually is highest level, the protein level. Yeah, they got the skin from different age, from 20s all the way to 90s, and compare the protein level. It's increased in the elderly. So, are we going to say it is a longevity gene or it's a anti-longevity gene? It's depending on how your judgment call, okay? But it's correlated with elderly adult. Yeah. So what is the P66? Actually, it's here, intact with, so this is a respiratory chain in the mitochondria. So intact with the cytochrome C between the complex 3 and the complex 4. And it's produced H2O2. So theoretically, when there's H2O2 production and intact with the cytochrome C may be released from mitochondria to cytosol, right? So this one of the intrinsic apoptotic pathway. Okay, this is one of intrinsic apoptotic pathway. So if the cytochrome C here released from mitochondria to cytosol, but turn out this pathway can regulate cell proliferation, can stimulate cell growth. So how we get into it? So that actually, I try to summarize what we are doing quickly because there are too much information. So we try to study how the P66 protein involved in the integrate the signal transduction, include the tyrosine phosphorylation, steroid hormone, and ROS, because we know tyrosine phosphorylation is essentially their protein are involved in growth regulation. So that growth factor receptor or oncogenic protein, that the tyrosine kinases. Okay, so tyrosine phosphorylation majority will stimulate cell proliferation. Then the steroid hormone actually regulate the gene expression, but now we know they have a non-genomic action. And then P66 protein apparently upregulated by the steroid hormone, androgen and estrogen. Then ROS we know involved in aging, cell growth, or pathogenesis or carcinogenesis. So all that is put together. We think this protein is actually a quite interesting one, especially related to the aging. Prostate cancer would not appear very rare to diagnose in the young adult, very rare. Only after age 60s. So aging is a factor that relates to prostate cancer very well. And then as we just see, the age population, normal population, the P66 going up. Then we just summarize what the P66 can do is involved in ROS production, involved in regulate aging or the oxidative stress. Protein is in the centennial old person. Tyrosine phosphorylation increases cell growth. Okay. And serine 36 is actually associated with apoptosis. So one protein can do two functions based on where is the phosphorylation, serine phosphorylation or tyrosine phosphorylation. Then in the cancer cell, actually P66 protein is actually elevated and correlated with the cell growth. Yeah, we have all the data to show that. So we decided to study the P66 protein in the prostate cancer. So the first thing first is, as I explained to you and the student, one of the students and all the person is, we need to have clinical correlation. When you are going to do a clinical phenomena, you need to have clinical phenomena. Okay, so we know in prostate cancer, high levels of oxidative stress is in the cancer cell, in the prostate cancer cell, but not in the benign tissue. So cancers in the prostate cancer cell, the oxidative stress going up. Then increase ROS correlated with the prostate cancer cell proliferation migration. And androgen could upregulate ROS in the prostate cancer cell. This has been established since 1992, well established. So this is a data shows published by others. Okay, the reference here is PIN is a precursor form of prostate cancer, and this is indeed the cancer. So in the precursor of the prostate cancer, this oxidative stress marker was not even is low level. When they become cancer, the oxidative stress marker biomarker, you can see much darker the staining. This immunohistochemical staining. So any question about immunohistochem? You understand? So is that we got a piece of tissue, we react with the antibody, primary antibody recognize the specific molecule, target molecule, antigen. Then we have a secondary antibody. They carry an enzyme, usually peroxidase. Then that will produce the color reaction. So the more of the antigen, you can see the darker of the color. Okay, so we can semi-quantify. And the PIN, as I said, this is a precursor form of the prostate cancer. Essentially not a cancer yet. And this is really the cancer. So additionally, not only immunohistochem staining, then they do analyze. They actually, this is a guanidine modification, the protein modification or the lipid modification. You can see in the cancer one, they're all going up. This is the cancer, this is the normal control. You can see the p-value less than 0.0001. So that shows support the clinical significance is in the cancer. The oxidative stress is going up, R is going up. Okay. Oh, so this is a progression of prostate cancer. So initially, when we have a cancer in the local area, then you can have most of the time. Now we have two approaches to treat it. One is surgically to remove, and some patients don't like to have surgical operation. They use a radiation therapy. So after the primary treatment, the cancer will going down, but then unfortunately, after a time, they will come back. So when they rebound, the tumor coming back. So this is the time we actually use we call the androgen deprivation therapy. So that means we reduce androgen from the patient. So that will suppress the cancer cell. So as I mentioned to you earlier, the prostate cancer depending on androgen. If there's no androgen, no prostate cancer. Okay. So androgen deprivation, we can suppress the cancer cell. But unfortunately, this chemotherapy, so the androgen deprivation, they are actually stimuli. There are two ways. One is surgical castration, and one is chemical castration. One is chemical castration. So we give anti-androgen to the patient. Then we can suppress the tumor growth 3 to 5 years. Then after that, you come back. That's what we call now castration-resistant prostate cancer. Classically, we call it androgen-independent or hormone-refractory tumor. But now the new term is castration-resistant prostate cancer. So the chemotherapy, one of them is the Taxol. As I just mentioned, the Taxol treatment can use serine 36 phosphorylation as a marker. Okay, so Taxol is one of the final agent to treat advanced prostate cancer. But again, this is only lasts usually about 1 year. About 1 year. When they become castration-resistant prostate cancer, the patient will succumb to the disease within a year. So this is what the cancer progression. So it's actually very similar in breast cancer too, very similar. Any question now? Okay. So this is our introduction. Now we try to study the p66 protein in the prostate cancer, right? So the first thing first is that really happening in clinical sample? Is p66 protein really increase or decrease in prostate cancer cell or no change? So the first thing first is we perform the immunohistochemical staining again. So we got the prostate tissue and staining it. Then you can see in the cancer cell, cancer tissue, the p66 protein indeed elevated. So we need to have clinical relevance correlation. Then that will be clinically significant. Okay. So those are from different patients. We got the tissue from different patients, and we have performed a statistical analysis. So we actually, one step further is we got the tissue from the same patient. Then the tissue containing non-cancerous area and the cancerous area. And you can see the cancerous area is standing stronger than non-cancerous area. So this is a base control. From the same patient, you can see side by side under the microscope. Indeed, that is the case. This is in the same slide. This is in the same slide. Okay, so p66 is going up in the prostate cancer cell. So then further is that related to the steroid hormone? Because we're talking about prostate cancer regulated by androgen. So is that really the case if p66 can be regulated by androgen? So we treat the prostate cancer cell with androgen following the dosage. You can see following the dosage treatment, the p66 protein going up, but not the p52, not the p46. And this is a cell growth correlation. So cell growth increased by androgen, p66 protein going up. So when we submit the manuscript to the International Journal of Cancer. That was a well-established journal. So one of the reviewer just put down, say, haha, someone finally knows how to regulate this protein. Yeah, you cannot believe it, put down haha. Yeah. So because even we knew it could be involved in longevity, but nobody knows it's involved in how can we regulate. We know the protein involved in free radical or steroid, but we don't know how to regulate. So androgen steroid hormone is actually, we show here androgen, but we also test estrogen example. So steroid hormone could regulate. So then that's why our working hypothesis, androgen could increase the P66 protein, then the cell growth increase. Then we turn out that we find androgen increase the P66 protein not through the gene regulation. It does not through gene regulation, but through, the stability of the protein by inhibit ubiquitination. So this actually was done by Dr. Kumar. Okay, he at the time in the lab and involved in this project. So we find steroid hormone actually increase the protein by stabilize the protein and inhibit its degradation. So that's what he did is with androgen is going up, and this is a proteasome inhibitor. It's a lysosome inhibitor. So we use a two inhibitor could increase the protein, but not the lysosome inhibitor. So shows this degradation pathway indeed through the proteasome. And the proteasome is actually responsible for ubiquitinated protein degradation. Okay, so are we okay? So now our next hypothesis is androgen through the P66 / ROS production stimulates cell growth and progression. So now we establish a relationship. P66 protein activate in cancer cell, and it can be upregulated by androgen, and then the activated P66 protein correlated with the cell proliferation. Okay, so there's a very strong correlation. So that was two other articles we published. Three of them shows we propose androgen stabilize the P66 protein, and it go to the mitochondria, interact with the cytochrome C, then release the free radical RS in growth stimulation. Okay? Any question? Okay. So this is what we try to establish the causal relationship step by step. That's why we are doing the experiment. The first we show here, androgen stabilize the P66 protein. Now we try to establish they go to mitochondria and then produce RS, then involved in cell proliferation. So this actually is we call the metabolic analysis or endocrinology. We perform the kinetic analysis. It's very tough. We perform the kinetic analysis. We treat the cell with androgen, DHT, and we harvest the cell at a different time point, 2, 4, 8, 16, 24, at different time point, and we run the Western to see the protein activation. So upon the treatment, we can see clearly the protein going up at 8 hours at a time, and then 16 hours. We can really compare with the control cell. And then the increase of P66 protein, we later on correlate with the cytochrome cyclin D1, is a cell growth, cell cycle regulator. And correlate with the PSA. We know PSA is androgen regulated protein. Okay, with the androgen, the PSA going up. So inhibit, reduce the androgen, the PSA going down. So PSA is a biomarker, important marker for prostate cancer treatment. When they're going up, we know the cancer coming back. So after the treatment, the PSA going down, the patient feel comfortable, will be released from the tumor burden. So this is a very good marker. So then we analyze the cell growth. So you can see the protein going up at 8-hour time, then we can see the growth regulation upregulate start at about 16 hours. Okay. So then this is a cell S-phase cycle. We can see the 24-hour, the cell number count, the cell number count. So we can establish a kinetic system. When the P66 going up, the protein going up, the RS production, this is RS production going up, then S-phase going up, then the cell number going up. So this indeed is the initiation factor. It's on upstream going down step by step. So kinetic is a very important one. So that's what I mentioned again is in the experiment, we do the dosage, we do the kinetic. So the dosage I just show androgen from 0, 1, 10 nanomolar stimulate P66 protein for the dosage. Okay. So here we do the kinetic. So this actually gives us the first conclusion here is androgen interact with a receptor, then they will stabilize the P66 protein going to mitochondria, have H2O2 production, then they can stimulate cyclin D1 for the cell proliferation. But androgen also at the same time can activate PSA secretion. So this is the pathway we actually interested in through the mitochondria. So it's really indeed the P66 interact with the cytochrome C. Is that really the case? So we harvest the mitochondria and the cytosol at the 8-hour and the 16-hour. Those are the time points in our early experiment. We see the P66 going up, right? So we got the 8-hour and the 16-hour, and we separate the mitochondria and then cytosol. And you can see the P66 essentially localize in, they can localize in the mitochondria interact with cytochrome C. Okay, so 16-hour in the control cell essentially, most of them gone. They go, we can see it in the mitochondria. Then they interact with the cytochrome C. So they indeed to get into the mitochondria interact with the cytochrome C in RS production. Then this one is actually PYK2 is a biomarker for cytosol because this protein would not go to mitochondria, only in the cytosol. So we did it, we hybridized the same membrane with the PYK2, try to show our separation is clean. We don't have a cross contamination. Your PYK2 also shows in mitochondria, indicating this band here, the P66 protein may be due to the contamination from cytosol. So you got to make clear your experiment, your procedure is actually meet the highest rigorous standard. Okay. So we find is actually the androgen could promote, stabilize the P66 protein into the mitochondria mainly. So of course, there's some of them still in cytosol. So this is, we do the IP. This is wholesale lysate. So we do the co-IP. You can see when we precipitate the P66 protein, we can see the cytochrome C co-immunoprecipitate. We call co-IP. Conversely, if we immunoprecipitate cytochrome C, then we can see the P66 shift. So that means if the two molecules form together, you bring down one, the second one should bring down, right? So then we do both ways. We bring down the P66 or we bring down the cytochrome C. We can see the pattern, interacting pattern. So both of them indeed form the complex in the mitochondria for RS production. Okay. Yeah. So androgen increase P66 and the cytochrome C interaction and in the mitochondria and RS production. Okay. So this is a little bit of complicated experiment we call rescue experiment. So if indeed P66 is responsible for androgen stimulation, now if we knock down the P66 protein, androgen stimulation should be gone, right? And then if we can put back the P66, then androgen stimulation should continue to go up. So that's what we call the rescue experiment. So this is a control. The non-wild type cell can stimulate by androgen. Then if we transfect the cell with a wild type P66 shift, even the control cell, the growth increase. The androgen stimulation actually dramatically reduce. But if we knock it down, in here we use a mutant form of the P66, and this mutant form compete out the wild type P66, so block the wild type P66 protein activity. So the mutant form actually block androgen stimulation. Compare the control cell, even though then even the androgen treated one just slightly increase. Okay, but if we transfer in here is, but this cell actually we can stimulate with the androgen for the cell growth. So back and forth, we try to do is P66 is really mediate androgen action. So this is a complicated experiment, and I think we just try to go through it quickly. All this experiment all together shows, yes, P66 is involved. If we knock it down, bring down the activity, androgen stimulation decrease. If we put it back, we can restore androgen sensitivity. So that's the key. Then we try to understand what is the signal transduction pathway, what is the downstream. So we perform the phosphoprotein microarray. Now we call the phosphoproteomics, but when we did it, we call the phosphoprotein microarray. So they changed the name. So we compare the LNCAP cell, the one androgen independent, versus the P66 subclone cell. Then the protein overlapping, those are the one could be the key player for the advanced cell proliferation. Okay, so yes, so this will say the computer show you the data, but you are the person going to determine who is the one you are going to target, because we cannot target all the protein. We cannot. We can only try one first and the two. Okay, one by one. So now we try to consolidate how the redox induced by P66 shift protein mediates androgen stimulation. How? What is the mechanism? So we know the tyrosine phosphatase actually activity can be inhibited by the free radical, right? So we can see we do have the subclone cell, this elevated expression of P66. Then we add the androgen, then growth. Subclone cell, we're going up. Oh no, this is knockdown, sorry. Yeah. So we have the androgen stimulation. So then which one phosphatase we are going to target in it? So we actually targeting to the PACP, the one I mentioned earlier, intact with LB2. Okay, intact with LB2. So on the different cell, now the P66 going up, then the LB2 tyrosine phosphorylation going up at a specific site. Only the tyrosine 1221/2, but not the other site. And then the signal can go down to downstream. So we find this is actually indeed this is a first world one. Okay, then we actually transfect the cell with inactive form of the P66. Then similarly, only the wild type can activate the ERK phosphorylation, but not the mutant. Then again, the phosphorylation exclusively at 1221/2. So then our working hypothesis, based on the data, our working hypothesis, the P66 protein go to the mitochondria, have RS production, inhibit the phosphatase. Oh, sorry, I totally mixed up. So this is phosphatase activity. With this S32, S31 here, the P66 subclone cell, they have higher P66 protein. Then when they have higher P66 protein, the phosphatase activity going down. Okay, so both of them, the phosphatase activity going down. But how can we prove the decrease of the phosphatase activity due to the oxidation? We said the free radical always inhibit, oxidize the free SH group, right? The cell group. So the phosphatase activity is inhibited. So what we did is the low activity here, then we incubate the cell lysate with DTT, DTT, the reducing agent. So when we incubate the cell lysate with the DTT, the reducing agent, then the SH group, the oxidized SH group, will be reduced back to SH group. Then we can regain the phosphatase activity. So indeed, after incubate with the DTT shown in here, then the activity going up. So indicating the phosphatase inside the cell indeed was inhibited by the RS. So therefore, the DTT can reduce SH group, then recover the phosphatase activity. And then this is what we got is RS inhibit the phosphatase, ERB2 phosphorylation going up in here and in here. Then they can go downstream signal pathway. Okay, so the ERK, the AKT, you can see the ERK actually also going up, the AKT activation too. So all this effort we just try to establish step by step, and this is really the final scheme. The androgen go to the nucleus and go to the cytosol, interact with the AR. Then the form the complex can stabilize the P66 protein RS production, then inhibit the phosphatase. Then the ERB2 can activate and go to the downstream. But if we treat the cell with the Casodex, it's anti-androgen. In clinically, we treat prostate cancer. Then when the androgen receptor is inhibited, then we can block the signal. Or we can use the mutant, same we block the signal. So we can do it step by step, okay, to get the result. So then at the same time, we also perform, I try to give you some example, metabolic analysis. This is, we analyze heme is actually a small molecule in the circulation inside the cell, regulate with RS, correlate with RS. So we find actually the heme level going up with from androgen sensitive cell to androgen independent, similar to here to here. So androgen independent cell is higher than androgen sensitive cell and correlated with the P66 protein. So this is a correlation. So we also find out the HO1 protein. You can see the cancer cell has HO1 standing. So again, the HO1 is going up in the cancer cell. So what we try to propose is HO1 is a reducing enzyme interact with P66 interaction inside the cell. So both of them, both of the P66 and the HO1 increase, elevate in the cancer cell. Okay. So this is what I mentioned to you. This really just a staining. The dye, the porosity orange 1 is a marker to see the hydrogen peroxide. So we can study in the cell to see where it is. Then in the AI cell, there are more RS, and the staining is higher. Okay. So this essentially just to show you in the cell biology and also what the dye we can use to do the H2O2 staining. And for example, the mitosol is mitochondria superoxide indicator. So these two markers actually very acceptable, well accepted by the people in the field. So we're talking about we might need to use a machine, but the machine is very tedious, very difficult to perform. Now we have very good tool dye we can stain in for the superoxide and also for the H2O2. And actually the data correlate very well. So this is a summary. What we propose is P66 in mitochondria can produce generate involved in the ROS production. When there's a moderate increase, the growth can stimulate growth factor receptor. The cell growth increase, the cancer cell growth increase and survival. When there's a substantially increase, too much, it might cause oxidative stress, they go to apoptosis. Then the ROS, as we mentioned, can activate NRF2 transcriptional factor. Then NRF2 can activate HO1 biosynthesis. Then HO1 could inhibit, is a reduction protein, can inhibit the ROS. Okay, so all this together, if ROS increase much, then we go to the cell proliferation, become a cancer cell. Okay, so that I think we try to go through it quickly because for the sake of the time. So those are the other experiment I try to show you is the steroid hormone interact with the protein phosphorylation, interact with the free radical. So this is really in the networking how we are doing. Not only say free radical go to free radical, how they regulate the, how we can regulate the free radical production and how the free radical could affect the downstream protein. Okay, so it's biology so simple. Now we know the cancer cell has elevated free radical ROS. Can we use? So there's a, as I said, there's a lot of tokens that use antioxidant, the fruits, vegetable. So this actually is a chapter we prepared way back long time ago in the book. So whether we can use for prevention, then turn out is the beta catalyst in the Chinese cancer prevention study, they find they show some efficacy. Okay. But in the Finnish group, they find out actually beta carotene actually increase lung cancer. Okay. Then in the woman study, they say no benefit. There's no benefit. So what is the beta carotene doing? So one possibility we need to know, there are different kinds of cancer. In here, they find out the beta carotene involved in the Chinese population. But this is a Finnish studies on the smoker, okay, the male smoker. So the smoker maybe have the additional ROS, the beta carotene cannot do. So this kind of major study is actually very difficult. Yeah. So until say more recently, 2009, there's a big selected trial, clinical trial, we call it select trial, use the antioxidant supplement against the prostate cancer. So they actually recruit over 30,000 patients involved in the select program. And they give them the select is a selenium vitamin E, okay? So the end result is, what is that? The end result is with the selenium vitamin C, turn out the prostate cancer instance actually increase, did not decrease, but increase. So compare with the placebo, the absolute increase in the risk of prostate cancer per 1,000 is actually 1.6 times for the treatment person. So the data actually opposite to what our dogma. Anti-androgen support should reduce cancer. But this is the case, it's actually resist. Okay. And another study shows is they put the tumor in the mice, and when the tumor established, they treat the mice with antioxidant. And then it's the same, the tumor grow better. The tumor grow better. So the two together, actually, we even don't know exactly what's happening. So one possibility, as I mentioned, is prostate cancer occur in the very young adult, age 20. Then when we recruit those patients, they usually are the 40s, mid-age person. The cancer already there. So actually we provide the antioxidant to those patients. Actually maybe try to counteract, try to reduce the tumor burden. So the tumor can actually grow better. Because when they are too high of the free radical, too high of ROH, actually cancer cells die. They become necrosis or apoptosis. And we find out in the P66 cDNA transfected cell, elevated expression, say 1,4, yes, the cell grow fantastic. But if overexpression by 5,4, the cell won't grow. They actually go to cell death, apoptosis. Okay? And then if those cells, we give them the antioxidant, maybe we can balance out, reduce their oxidative stress burden. The cell become more viable, so the cancer incidence increase. So then the possibility is we might need to give the patient at the early stage, say age 20s, and really a prolonged study. But unfortunately, that kind of the study is cost a lot. We don't think there's anybody can avoid, especially in the US. We cannot avoid anymore to do those kind of experiments. Okay, so finally, I thank you. I will quickly go through the data, and then I need to also acknowledge people, okay? Especially Fun, as you saw her here, she's my wife and also my boss, okay? She controls all my budget, all the economic, financial. Actually, be honest with you, sometimes I even forget my password, how to get into my credit card or something. She's the boss, regular everything. I remember, yeah, Suraj, Dr. Kumar, yeah, he was involved in the study. We have many people involved. So actually, one of the very talented, my former associates, Dr. Tsukasa Igawa, yeah, he actually is the person suggests to me we study the P66 protein in the tumorigenesis, but not in the apoptosis. So initially, in the early phase, everyone thinks P66 interact with the cytochrome C, go to apoptosis. So most, I would say over 99% of study on this protein are in apoptosis. Then Dr. Igawa, he was involved in the project. One day he come to my office, he said, Dr. Lin, can we study cell growth, but not cell death? I said, oh, it's a good idea. He said, yeah, I've been doing the cell death. We should be different from others. I said, okay, it can be different from others, but it could be also a failure. We need to understand that, okay? And we tried bingo that established our last 10 years, over 10 years of research going on. So he's currently professor and the chairman in the urology department. He was a urologist in one of the Japan university. So it's very important, but I not say, I give him as an example is when you are doing the experiment, you are the one, you have hand-on experience, you know better than your advisor. Your advisor will be as me sitting in the office, just typing all day long. We are the armchair scientists. We are not we will be, yeah, unfortunately, we don't like it, but this becomes the case. We become the armchair scientists. So you are the person in the lab doing those wet lab experiments. You know better. So you can make, you need to, all the creative actually coming out from you, not from us. Okay? So, yeah, I always intibate to him and his suggestion. Okay. Who is him? Dr. Kumar, okay? So this is 2009 when he joined our lab. Yeah, so this is my boss. Okay, she actually in the lab helped me to taking care of the lab, like a lab manager, everything. All the new person, even Dr. Kumar was, she taught him how to take care of the cell culture, the basic beginning, over and over. So actually you can see, actually Dr. Kumar and her, they are very close. They know each other very well. Yeah, because they actually interact daily, every day in the lab, but not me. Yeah, but not me. They don't like me, I can tell you that, because they think I'm a crazy guy, okay? So they actually interact very well. So this is a Satendra, this is a Nagendra. They all are postdoc. This is another postdoc from Taiwan, and this is a urologist resident doing the research in my lab. So urologist, he is a clinical person. So this actually, yeah, at the time we have the people. So you can see everyone, they're actually working very hard. That's what create our lab. So when I was young, I do have hair, okay? I do have hair. As I say, my father now, he's 100 years old. He has more hair and darker than me. Yeah, he had more hair and darker than me. Don't ask me why, maybe I work too hard. Yeah, this is my family, and I need also, yeah, my son is actually a urologist now in University of Wisconsin Medicine. And my daughter is an internal medicine nephrology for kidney cancer, kidney study in MD Anderson Cancer Institute. So actually, the reason I show them is I always in the lab. I barely go home. 24/7, okay? I go to the office in the early morning, go home late, and every day. So until when my son was going to college, because the college, they will go away from the home. So he told me, he said, Daddy, do you know, I'm leaving. He told me when he was 11th grader. I'm leaving in 2 years, then I will never come back. Of course, he come back as a guest, but not stay with us. He said, I'm going away in 2 years, and you are still working so hard. I barely see you. Are you sure that's what you would like to be? So then I start to cut down my office hours from 24/7, say, to 24/6. So Sunday I stay at home. Okay, at least I can stay home with him. Then to the last year, yeah, he said to him when he was in the 12th grade, so the high school 12th grade, he said, I'm leaving next year. You only stay 1 day. So I just bring my laptop, everything, all my file to home. I'm working at home. Then when he wake up in the morning, if he saw me in the home, he said, you are not going to the office. I say, no, I'm not going. I stay home with you. And he will go out. He will go out to pray, but he is happy I stay home. Yeah. So yes, as a research scientist, I have a lot of sacrifice to the family. I need to admit to that when you are really, push yourself. So that's what I say. She's the boss. She takes care of the 2 kids. And luckily, both of them actually grown up and well established in their respect field. And when they decided to go to medical school, I can tell you the truth, I was shocked. I asked them, are you sure you like to go to medical school? The reason is, yes, I can go to medical school for medical doctor, but I run away from medical school. The reason is I cannot do the gross anatomy. I cannot do the cadaver, to do the gross anatomy, cut a patient, a human being. I cannot. Okay. Just subconsciously, would not allow me to do so. So that's what I give up my medical school, went to the PhD program. Even I graduated from pharmacy school. But I did finish actually the first year of the medical courses. So that actually also helped me the background to do the cancer research, because I should be very proud of myself. I know so much in every different aspect. I know the clinical, I know the pathology. I can talk with the clinician, no problem. I can talk with basic scientists, no problem. But still, it's a lot of sacrifice. But again, it's not a bad job. The pay is reasonable. Then we receive the reasonable pay, and then we learn the new idea, new knowledge. Then at the same time, your result could benefit to other human beings. So what other job is better than that? Yeah, you might think about the AI or something. You are interested in AI, yes, go for it. Not to say medical school or research is really the way you need to go. It depends on your interest. Then go for your interest. Very, very important. So for example, my two kids, they go to medical school. We did not encourage them. We did not. Because really not what we can tell. When you grown up, your partner cannot make the decision for you, right? Similar, I cannot make decision for them. Okay? So they make their own choice. But the specialty is the same, make on their own choice. So again, I think I'd like to thank you for your 1 week patient, persistent or stubborn with me for my Chinese accent English, okay? And hopefully Dr. Kumar will give you the slide and with my with all the recording and everything. And you can try to understand what the Chinese language. Yeah, actually this morning, for example, I went to other college to give a talk. I learned actually most of you have very few chance to encounter international people. But one way I encourage you guys to go outside is because this has become a global village. Okay, it's become a global village. You can see people going everywhere. It's the travel is so convenient. You can see any kind of people. So go outside, broaden your vision. That will help out a lot for your career. Doesn't matter which way you want to go. Doesn't matter. Okay, thank you. Any question? Question? Any question? I know you try to go for dinner, lunch. I'm hungry too, but yeah, no, any question will be fine. Any question? No? Am I clear? Oh, this is not a question, okay? This is a request, it's not a question, but I'm talking about the question first. What is the question? No question. Yeah, so I try to say is, yes, the similar transaction like your traffic. So for example, this morning, say after my presentation, my meeting with the college student, when we come back, we find there was a traffic jam. Then Dr. Kumar, not Santoshi Kumar, another Dr. Kumar. Okay, I see, right? I see. Then we quickly turn around from the other direction. Okay, so this is a way